Kia ora koutou. I'm coming to you from New Zealand, and I'm going to take a bit of the surveillance side of things, since that there will be a lot of talks coming after me that will be talking about the technological side and how we actually do some of that sequencing. So that's what I'm going to focus on. So ESR is within New Zealand the National Reference Laboratory, and we deal with all kinds of things from health to food and environment. And today we'll be speaking mostly about the public health side of things. So there's a bit of a spoiler in the title here. It's what we wanted to do, and it didn't quite work out how we envisioned it, but I'm going to just kind of take you through the story of how we geared up to be doing sequencing of the virus and what we've been learning from it along the way. So we really wanted to try to give a shot at real-time genomics to support public health with their contact tracing, to assist their understanding of the outbreaks, and to also perform phylogenetic modeling to support decision makers about control levels and border closures, etc. What do you need to get that? Of course, you need rapid referrals, rapid sequencing, rapid data sharing, and rapid interpretation and follow-up. The middle two there are sort of under our control, since that we control the sequence technology and how we interact with that data. The other two are, of course, parts of a larger system that is a nationwide endeavor, and we'll come back to that a bit later. So we were getting ready. News started spreading about the virus, and the people from the Arctic network did an awesome job of getting a primer design up and ready very quickly when the genome was sequenced from the Chinese strain. Luckily, we were in a position that we had invested in capability and capacity to perform sequencing at scale. So we reached out to long-term collaborators in the UK, Joshua Quick, who was part of that network, to send us some primers. So on January 28th, they shipped that to us, and then shipping was still pretty okay. So we received that in a timely fashion. And because all of the protocols are open source, we could just check them, get our bioinformatics set up, and you'll probably hear more about that in later talks. So we brushed up on our next strain usage, because we typically deal with bacteria or humans, not necessarily viruses, but NextStrain was one of the platforms that had been used in Zika and Ebola. So we thought that's probably a good way to go. And GISAID submissions, and we got the nanopore MinION ready because we wanted quick turnaround times for our sequencing, and they're just a bit quicker than running a full Illumina run. So the first positive case was detected in New Zealand on the 1st of March, and it was indicated that there was a travel history from the Middle East. And we had the genome running on March 1st, so that we thought that we had that in hand pretty quickly. We had some low coverage regions. As some people know, the early primer scheme, there were some dropouts, so we left it running for a little while. And this is actually us looking at the data. So our comms people actually got us a shot where we're actually looking at the data. And the good thing about viral genomes is that you can actually look at the whole genome in a text file. So it's one of the few times that you can actually do that, and not just for a comms photo. So we had that up on NextStrain on the 6th of March, and because of large international data sharing efforts and sequencing efforts, we actually saw that it grouped with some other samples, and for some of those it was indicated that they had a similar travel history. So there's 4 ones here circled. They all had travel history to Iran, and the others that were not available, but very likely they came from that same region. So that all seemed to be working well in terms of the setup and the sharing capabilities. And very luckily, we were able to hook up with some people in New South Wales that are a lot better at phylogenomics than we are and understand a lot more about viruses than I do. So Edward Holes and JS, as he's called in the community, we got together and we were able to write a short story about an emergent clade of the virus that seemed to be coming from Iran. And the interesting bit to note here is that we were only able to do this because there was a large international data sharing effort. because Iran itself was not necessarily performing sequencing. So only by piecing together the travel links from those genomes and linking them up with the mutations that are there at the trunk of that tree, that we can confidently say that this is likely from an Iranian source. Then reality happened. Rapid ramp up of test capability. We just heard about some of the logistical challenges that brings. Of course, there's also the challenges of people that all of a sudden have to work in shifts while they might not necessarily be used to that around the country, because New Zealand took the test and test approach. So this is a distributed system under pressure. We got border closures. There were disruptions to shipping when we went to full lockdown levels. And many people working on this were doing an excellent job from home. But if you have children at home, you will probably relate to this saying that it's being at home while trying to work rather than the normal working from home that you might be used to pre-pandemic. So it's not quite normal circumstances. So what we were able to do thus far, we've sequenced nearly 250 genomes. Most of them were good quality, and some of them are a bit weird. Our current cutoff is less than 3,000 ends, so missing 1% of the genome is okay. This mostly has to do with amplicon dropouts that sometimes still happen with fragmented material, or sometimes with very good material for weird reasons. We use a combination of nanopore and Illumina sequencing, nanopore for the really rapid samples, and Illumina for the larger bulk of samples that we want to do. And we use the Arctic V3 primers, and as we call them, New South Wales primers. So those are the longer amplicon primers of about 3 kb that we use for fragmentation and then running on the Illumina, because we think that slightly gives us slightly nicer coverage of the genomes. We focused a lot on samples with an epidemiological impact, especially cases where there were unclear infection sources and the larger outbreaks and potential transmission chains within the country. The epidemiological team has been prioritizing those and getting couriers running around the country to try to get them to us in a timely fashion so that we can provide genomics insights into those. And now that case numbers are luckily nice and low, we can sequence them as they come in, and with the distribution system being back up and running, that's looking a lot better. So some things that we have been able to get from the data, and to some people that might not be surprising, but I still think it's worth noting that this is truly an added value to the epidemiological surveillance that is typically happening. For those cases where there was a source unknown or unclear infection route, for many of them, we'll be able to provide hints either by the region of travel that people had to look at, which then helps to look at flights or things like that, or to other known cases within the country that can then be followed up. And then there's also larger outbreaks that are easier to determine epidemiologically because people went to the same event or were in the same region at a certain point of time. But even in those cases, we often were able to link additional cases to those outbreaks by genomics that might not have been found directly from EPI. And then when they go back to people, they actually figure out that they were in that area. They just hadn't remembered that bit. And we can get a more clear understanding of how that outbreak has been spreading. And in some outbreaks, there can be different transmission patterns. So they might be epidemiologically linked because there was maybe the indication that they came from the same area when it was travel introduced, or that they were in the same area, but actually genomically there might be separate strains, which helps to understand and track the spread of the virus. We got genomes from all over the place, being a touristic country. That was sort of expected, but it's also reflected in the genomes. It's just a B4 clade that we don't seem to have any genomes from, but it's a fairly rare one. But all the other ones are represented, and with more genomes coming online, that's probably the picture that we expect to continue. Another thing to note is that islands seem to be doing well, and we've heard comments earlier about the border closures. That's something that we can control, which is very helpful. So I've highlighted 3 well-known island nations here. And you can see that we're all heading in the right direction. Very fortunate for us. But it's also something that we wanted to take a look at from a genomics point of view. And luckily, within New Zealand, we have a very excellent group at the Center for Computational Evolution, led by Alexi Drummond and David Welch in Auckland, who do a lot of phylogenetic modeling and are the authors of algorithms like BEAST that some of you might know. And they actually took a look at the genomic diversity and where the genomes were coming from, and they compared that to the actual arrivals into the country. And what we can see is that even from the genomes, we can recapitulate the picture of what happened to our borders. So in the yellow line, you see the actual travel, and in the blue line, you see our genomic, the genomic reconstruction. And you can see that when the borders closed, we see a rapid decline in that diversity, meaning that we went from imported cases to household spread or community or outbreak types of events. So it's really interesting that only from a limited number of genomes, we can already start to see that in the genomics as well. And then there's also the effects of lockdown. So a lot of people have said that lockdowns are effective, but they're fairly crude measures. And are they needed? Were they in time? Those types of questions. So another institute within New Zealand, which is very heavily on the modeling side, Te Pūnahu Matini, has done a lot of modeling for the government and their response. And this is one of the graphs that they produced to look at when lockdowns would enter effect and how that would then do for the outbreak and the numbers of cases. And with the lockdown measures in place, they predicted that the R value, the effective value, should go down. So one person doesn't affect at least one other person, but less than one, which means that you're flattening the curve. And again, New Zealand took a lockdown hard and early approach. So here we see some data on whether New Zealanders also listened to that directive. So we went to what they call lockdown level 4, which means only essential businesses were operating. And with companies like Google keeping track of where everybody is, we can see that New Zealand was very obedient, and indeed it was a strong lockdown. So there were significant reductions in people spending time in recreational or parks. And if you look at the graph on the right-hand side, people were staying at home. So that's the green bar that goes to the right-hand side of the graph. For some reason, all the Danes started to go to their parks, which is an interesting observation, but not for the comment here. We also took a look at the genomics again to see if what patterns we saw for the reproductive number. Again, we're only using a small number of genomes for this, but if we exclude all the known travel introduced genomes, and we account for that in the model, we can see that prior to the lockdown, there was a R that was greater than 1, even within New Zealand. So there was a potential for an outbreak. We think that this R was still fairly low because it might have been summer, but there's still a lot of people working on the seasonality. So we hope to be able to say something more about that in the future, but it's an indication. And what we can also see is that actually in alert level 4 in the genomics, we can also see that that reproductive number is indeed below 0.5, meaning that we're really flattening that curve quite effectively. And this was provided again by the Center for Computational Evolution by Fabio Mendes and Jordan Douglas, two fabulous people working on that sort of data. So we wanted to do this really real time, and we're still striving to get there. Luckily, there's been a good project started now led by Colin Simpson and David Welch from Auckland and Victoria University to get this into a more real-time setting. So we're looking at not just genomic data, but also cellular data, travel data, weather data, and all the other data sources that might be coming in more real-time to get these surveillance systems up to a level where we could do all of these interpretations integrated. Then a brief in other news before I'll stop talking. That is that we will plan to do some direct RNA sequencing, since that between Otago University and ESR, there's now several strains that have been cultured. And apart from the direct RNA sequencing, that's also for assay validation and those kind of methods. And that we've had some first preliminary success in isolating it from wastewater. This has been something that in countries with high caseloads, they have been proving that it's a way that you can monitor the spread and the sort of magnitude of the outbreak. But yeah, with the lower case numbers that we had within New Zealand, we still detected the virus, but more work is clearly needed to make this a more effective long-term monitoring tool. It really takes a team to do all of this. So this is my acknowledgement slide of the large number of people here. And a big thanks to the public health units and diagnostic testing labs for all the hard work that they're putting in to get these positive cases and to follow up with people and ask all the difficult questions, especially seeing that it's still a serious disease that we're dealing with here. And this is provided with the epidemiological work and the outbreak focused work is done for the Ministry of Health. And we just heard from our Ministry of Business and Innovation and Employment that they will be funding the sequencing of all positive genomes in New Zealand. So we'll be endeavoring to complete that work in a timely fashion and provide a New Zealand view of how this pandemic happened.